In this module, we will discuss the activation of downstream signaling pathways. That the pathways which are downstreams, how they are activated to carry out the cell's activity. And in this case, the receptor protein tyrosine kinase, RTKs, they are autophosphorylated on one or more tyrosine residues. It is very important to discuss and to keep in mind that the tyrosine kinase, receptor protein tyrosine kinase, they play an important role during the activation of downstream signaling processes. Now, a variety of signaling proteins, they are available, for example, SH2, PTB domains, and they are present again in the cytoplasm. Remember that these receptor proteins, they are present in the cytoplasm of the cell, inside of the cell, not outside of the cell. So how these formation of signaling complexes, it results. So this is it, that the receptor activation result in formation of signaling complexes in which again the proteins SH2 or PTP containing signaling proteins, they bind to specific autophosphorylation sites which are present on the receptor. This is very important that we should remember these two proteins as well, SH2 and PTB, which are there and have certain conformation there. Now, the group of signaling proteins which we can distinguish, they are, as a matter of fact, two types, rather three types. One are the adapter proteins, docking proteins, and transcription factors, and of course, enzymes, which are the protein molecule. So, there are four types of these signaling proteins, which should remember: adapter proteins, docking protein, transcription factors, and enzymes. So we should remember these, that they are the one who control the activation of the downstream signaling. Now, what are adapter proteins? They function as linkers, जोड़ने के काम आती हैं, that enable two or more signaling proteins to become joined together as a protein of signaling complex. जिस तरह नाम डॉकिंग अडाप्टर प्रोटीन है, वो ये है कि वो अडॉप्ट करती हैं, वो जोड़ देती हैं उन कॉम्प्लेक्सेस को. अडाप्टिंग प्रोटीन्स वो कौन कौन सी हैं? SH2 domain and one or more additional proteins where they interact in the domain with each other to control the cell signaling devices. Now, for example, in this case, what I have been talking times and again, that these are the tertiary structure of adapter proteins. Mind you, the proteins, they have got three structure: primary, secondary, and tertiary. And tertiary structure are the one which are very important in the biological system because they change the conformation. वो अपनी माहियत को बदल देती हैं and to control the cell signaling. तो आप इसमें देख सकते हैं GRB2 consists of three parts: 2 SH3, दो SH3 के proteins and domains, and one SH2 domain. So they will make GRB2, which will make the signaling of the cell possible. Now, docking proteins. जिस तरह नाम जाहिर है, docking क्या होती है? These are the proteins as IRS, and they supply certain receptor with additional tyrosine phosphorylation sites. Now, docking proteins contain either a PTB और SH2 डोमेन जिस तरह हमने पीछे देखा है, एंड ए नंबर ऑफ टायरोसिन फॉस्फोरिलेशन साइट्स। ये डॉकिंग प्रोटीन है। डॉकिंग प्रोटीन का मतलब ये हुआ कि वो एडिशन, वो डॉक करती हैं उन प्रोटीन्स को, वो जोड़ती हैं एक दूसरे के साथ, जिस तरह स्पेसशिप नहीं डॉक करते स्पेस में, दैट वी हैव द स्पेसशिप हैज डॉक्ड, सिमिलरली दीज प्रोटीन दे हैव डॉक्ड with each other and connect this. Now, transcription factor, आपको पता है कि सेल में DNA में ट्रांसपेक्टर फैक्टर that belong to STAT family, it plays an important role in the function of the immune system. क्योंकि ये प्रोटीन्स हैं, there will be immune system as well developing at the same time in the biological cells, so they will control that. And these transcription factor will form dimers. Transcriptor factor हमेशा क्या बनाएंगे? डाइमर बनाएंगे, दो, क्योंकि वो मोनोमर में नहीं हैं. Dimers, but not monomers, they are made and they move to the nucleus where they stimulate the transcription. देखें ना, डाइमर जो हैं वो किधर जाएंगे? सेल के न्यूक्लियस में जाएंगे. और सेल का न्यूक्लियस किधर होता है? अब मैं आपको बताऊं या आप जान गए होंगे कि दैट इज इन द साइटोप्लाज्म, एंड दे स्टिमुलेट द ट्रांसक्रिप्शन ऑफ स्पेसिफिक जीन. जो स्पेसिफिक जीन्स हैं, जो पर्टिकुलर जीन्स हैं, जिनकी तरफ वो सिग्नल आ रहा है, वो उनको स्टिमुलेट करेंगे, उनको एक्टिवेट करेंगे टू इन्वॉल्व इन द इम्यून रिस्पॉन्स. उनकी जो इम्यून रिस्पॉन्स है उधर. नो सिग्नलिंग एंजाइम्स, the fourth one and the last, they include protein kinases, protein phosphatases, lipid kinases, phospholipases, and GTPases activating protein. They are making the signaling enzyme. That is, they will, as a matter of fact, signal and transduce the signal which cell should receive. And at the same time, I think I should leave a question with you, that where these signal enzyme will be in the cell again. So you know the answer. Now, when equipped with SH2 domains, these enzymes associated with activated RTKs and are turned on directly or indirectly as a consequence of this association. तो क्या होगा कि ये SH2 डोमेन्स ये एंजाइम RTKs के साथ एसोसिएट करेंगी एंड दे टर्न डाउन डायरेक्टली और इनडायरेक्टली एज कॉन्सिक्वेंस ऑफ दिस एसोसिएशन. व्हाट वी मीन बाय टर्न डाउन? टर्न डाउन का मतलब ये है कि उनके कन्फर्मेशनल चेंजेस, उनकी जो चेंज है, जो टर्शियरी स्ट्रक्चर का चेंज है, वो इस तरह हो जाएगा कि सेल सिग्नलिंग जो है वो पॉसिबल हो जाएगी. Now, enzyme can be regulated directly for phosphorylation, remember, because enzyme molecule is a catalyst. So they can be regulated by phosphorylation, ATP के साथ phosphorylation, and so on. So these are the major factor in this chapter we have studied, which control the downstreaming regulation of the cell signaling.